I don't know why this bar shows up over my other bar. That's what the problem is. So that's okay. There's more than one way to skin a cat. From the current slide. There we go. So transposons, these happen naturally in nature, right? They were discovered by Barbara McClintock. You don't need to memorize her name, but I think it's cool. It was a female scientist, right? Isn't it all guys? It's a good mix. And she discovered it in maize corn, right? Maize corn has all those different colored kernels, and I was like, well, why is it different? It's all from the same plant. It should all be the same. But genetically, you know, genes can move around, and that's what was happening. And when they do that, they can disrupt other genes, and that's what you see happening here, is gene B is being disrupted. So I always, too, like to give you guys, you know, a real-world example, real stuff that really happens. So this is related to some of the stuff that I did even as a graduate student working on my master's at Southeastern, is that we would use bacteria, and we would manipulate them genetically to be able to do certain things. What I was doing was actually inserting a sequence of DNA from a frog into the bacteria, and I was just using the bacteria to copy that DNA for me so I could get lots of copies and then run it on the sequencer. And so I was able to sequence a segment, a gene of a particular protein important to our immune function, but in a particular frog. And I did that for 3 different frog species. So in that technique, one of the things that we do is we have these markers, we have these abilities to insert DNA, right, into plasmid, circular pieces of DNA that these bacteria have. So these are specially designed bacteria for allowing us to use them basically as little machines and copy DNA for us. So one of the ways we can know, because you can't just look at DNA under a microscope, right? Although they do have high-powered microscopes now, but they're still not going to tell you the DNA sequence, right, to really know that you were successful in what you were trying to achieve. So certain genes give cells certain abilities, right, give them a particular phenotype. So these bacteria come with this particular gene that allows them to split a substrate we give them. It's an enzyme that they make from this genetic information. And they'll split this substrate we give them from a colorless molecule, and when it splits it with the enzyme, it turns blue. So the bacteria grow blue in their natural state, the way they come from the manufacturer. What we do is we insert into them a plasmid that contains an enzyme that can destroy an antibiotic. And I just chose penicillin because that's an antibiotic name that most of you guys are familiar with, and you know what it does. But it's not actually what I did in my research. Attached to that is my sequence that I want the bacteria to copy for me. And so we would insert this, and it is coded so that it will insert into this other gene. It will disrupt this gene. They will no longer be able to make this enzyme because that gene has been split into 2. And when the enzymes in the cells go to copy it, they don't know it's been split. They won't jump from one section to another. Enzymes don't do that. So this one has been essentially disrupted, or what we call being knocked out. So that cell has lost that phenotypic ability. It will no longer make this enzyme. So it can no longer take that substrate I give it and split it and turn blue. Instead, they're going to stay the normal color most E. coli are, which is white. Also, they will only grow on the special media I'm using because it contains penicillin. They'll only grow on that plate if they took up this genetic information, because now they're producing an enzyme that will destroy the antibiotic. So I'm able to even select, not only by color, but by the fact that they've genetically changed now that they're antibiotic resistant. And they will grow on the plates that I use that only allow ones that are resistant to that antibiotic. And of course that bacteria before I changed it was not resistant to that antibiotic.
So, not to get out too much off subject, but as far as penicillin cheese, is it considered an antibiotic?
No, the penicillin fungus that grows in your cheese to make the different types of blue cheeses is not in a situation where it's really fighting for nutrients, so it's not producing penicillin. Remember, penicillin is its protection, its chemical warfare that it uses to fight other organisms particularly bacteria, gram-positive bacteria. So under those conditions, it's not producing the penicillin. Right? So people with penicillin allergies and stuff can eat those cheeses and not get sick. Because even though it's the mold that makes penicillin, it's not making it under those conditions. Make sense? Excellent question, actually. Yeah. So we have to actually stress bacteria out in the lab, stress out that fungus in the laboratory setting to get it to produce the penicillin. Right? Put it in a stressful situation. And then they purify the penicillin from the culture. So here, right, I'm able to know that we made these changes, right? And the important thing here is that we're using transposable elements. We're using segments that will insert into other segments and disrupt them. Right? So although this happens in nature, like Barbara saw in the maize corn, we can also cause this to happen in the lab. And in that case, it's referred to as an induced mutation. Right? We're causing it to happen. Other things that can damage DNA is UV light, right? And that's why it's important that we protect ourselves to exposure of ultraviolet light. It literally can distort your DNA. But it's not very far penetrating, and that's why you can wear things like sunscreen, and it helps protect you, right? A very thin layer, right, can help protect you from the penetration of UV light into your skin cells and help protect your DNA. But you got to actually wear it, right? Or you got to limit your exposure to sunlight. Any of you guys seen Dean Gruber, the Dean of Math and Science? He has a beautiful nose. Not really. He's had 2 or 3 surgeries, and this even happened to my dad, because my dad was a baseball coach too, right? So spent a lot of time outside, unprotected from light, ended up with cancer on his nose, and had to have surgery to have it removed. And the same is true for Dean Gruber. If you see him, his nose is very distorted. And it's because, he was a coach for many years, spent lots of time out in the sun, didn't protect himself from that sunlight. So what is actually happening at the molecular level to your DNA when it's being exposed to UV light is that, remember the A's, T's, G's, and C's, those nitrogenous bases that make up the code in DNA? If there are 2 thymines next to each other, instead of hydrogen bonding like they're supposed to with the adenines across the strand from them, they will covalently bond with each other. And this is a stronger bond. This is where they're sharing electrons. And the bad news with this is if you notice the DNA backbone gets a buckle in it, right? And that's really bad for the enzymes that read our DNA. Because as I said before, right, your DNA is that double helix. This backbone, this sugar phosphate backbone that attaches the nucleotides together, is almost if you imagine a spiral staircase with a banister, right, or a hand railing, right? The enzymes literally attach to that hand railing and slide down the molecule and read it. Well, what happens when it gets to a bump? The same thing that happens where, places where they don't want kids sliding down, like escalators and stuff, what have they done? They've put in obstacles in their way so that they would fall off or not do it altogether. So this is bad news for our enzymes because when they get to those bumps, the same thing's going to happen. They're going to fall off. And they're not going to be able to read that DNA. And you need it to be able to read your DNA, right? That's your information. That's your command center. You don't want this distorted. You don't want it unreadable. So the good news is, just like for microorganisms too, we have enzymes that can repair this type of damage. But if you have too much damage, right, too often, it's more than the body can keep up with, right? And that's when it can lead to problems. The same thing with microorganisms, right? When we overexpose them to UV light has been used a lot as a disinfectant. The problem is it can't penetrate very far, right? So like if you're disinfecting water with UV light, it has to be very thin ribbons of water that are being passed by the UV light for it to be able to penetrate into any of the bacteria that may be present in that water or microorganisms. And, exposing them to this type of radiation, this ultraviolet light, will cause this type of damage. And if it's too extensive, it could lead to the death of the cell. So protect yourselves, right, from this type of damage.
Oh, so the UV lights that we have for the toothbrush case really work then?
Yeah, it can help. Yes. Yeah, I didn't know they had that.
Yeah, I saw one.
Yeah.
Like gadgets and stuff. Yeah.
Yeah.
Teeth cleaning, you know, little teeth cleaning, people walk around with little teeth cleaning thing.
Yeah, that's a little extreme. I wouldn't expose myself. I wouldn't expose my body to that. But, you know, other things that, you know, are not your body. Like tanning beds, worst invention ever. Don't do it, y'all. Like, if you want to look tan, right, go get the spray tan. Or just, you know, white is beautiful. Truly. Right? And then those of you guys that produce more melanin than us white people, right, that does not mean you can't get cancer. You're just less likely. You have a little bit more protection than we do, right? And you're lucky in that aspect. But, you know, it can still happen.
Yes.
Yes, you can get sunburned.
Yes.
Yeah, it can happen. It can happen. And I have a friend that was Asian, and she was, you know, when I was a kid, I didn't understand these things, right? You know, the different levels of pigmentation and everything, and she was putting sunscreen on. I'm like, why are you doing that? She's like, I don't want to get any darker. Because in that group of people, right, actually being dark pigmented is not favorable or something. I didn't know. I was like, oh, okay. Whatever. And then I found out later on, I was like, well, it was a good thing that was, because it helped her out, because she's less likely to get cancer because less exposure to the sunlight, skin cancer anyways. Right? So, yeah, we all of us, no matter your skin tone, really should protect yourself from ultraviolet light. Right? You have a little bit less of a risk factor if you have lots of melatonin, if you're dark pigmentation, but it can still happen, and yeah, you can still get damage to the level of burning from sunlight. All right, so for the next couple, for this, now we've been talking about mutations, right, that would happen, say, in the mother cell and be passed on to the daughter cells, and we said, you know, this can lead to lots of different outcomes. It could be good, it could be bad, it could have no difference at all. We had a couple of terminologies in there that you guys need to learn. So now we're looking at horizontal, where it's not going from mother cell to daughter cells. This is something moving, say, from one cell to a cousin or somebody who's not even related, right? So, like, for instance, going from Shigella to E. coli. And we believe this has happened, this horizontal gene transfer. Now, which one of the three mechanisms that we're going to talk about, we're not sure which way it moved, but there is a toxin that's present in Shigella called the Shiga toxin that's also present in a pathogenic strain of E. coli called Shiga toxin E. coli. The most common strain that has that characteristic is E. coli 0157. If you've heard that strain designation, right? It has that toxin. It's the same toxin that Shigella has. It's the Shiga toxin. So it probably got it from that bacteria, right? From that species, it was transferred over to E. coli, right? And not all E. coli have it, right? Just that pathogenic strain. So that's a real-world example of horizontal gene transfer. How it actually happened, we don't know which one of these mechanisms. There's 3 different mechanisms that fall under horizontal or lateral gene transfer. There's DNA-Mediated Transformation. And of course, all these involve DNA, but there's other transformations out there. But typically, most scientists usually just say transformation. And then you'll notice the next one says transduction. And those words are very similar, right? They both start with trans. So you can sometimes get them mixed up. So hopefully I'll give you guys some strategies to not mix those two up. And then the third is conjugation, which you've probably heard before, right? And there are other organisms that do conjugation. Another name for this is sex, or bacterial sex, because all sex really is an exchange of genetic information, right? Even for humans, down to the very basic level, it's DNA from a male to a female. All sperm are mobile DNA. Of course, it's kind of a cool process of how all that happens, right? But down to the very basic essence of it, right? Sexual reproduction is all about exchanging DNA. So the same thing with bacteria. They don't have to do it to procreate, right? They have mother cells. They can just do binary fission, copy each other, asexual reproduction. No big deal. No need for someone else. But they can exchange information. They can join up with each other and exchange DNA. Right? That would be a form of sexual reproduction, known in bacteria as conjugation. They're just not required to do it, but they do it. Transformation, transduction, as I said, they sound kind of familiar, right? But as we talk about transformation, probably one of the best ways to like really get that term in your head of transformation is transforming, changing. And thank goodness for transformers, right? My son's favorite one is Bumblebee. There's someone who drives that Camaro around this city. We've seen it a few times. My son is convinced that it will turn into Bumblebee. Told him numerous times, no, it's just a car. That's make-believe. There isn't any such thing of a car actually being able to turn into a robot. But who knows? Someday it could happen, right? So I should really say to him, as of right now, I'm pretty sure it's just a car. Right? But that's a dramatic change. That's a transformation, right? Transformers transform, right? From a car to a robot and back again. Same thing with microorganisms, right? If they get a lot of genetic information, this genetic information allows them to make stuff, to basically transform into something they weren't before. So in the microbiology lab, we actually do this with E. coli. We give them DNA from jellyfish that is a fluorescent protein that fluoresces under UV light. E. coli usually do not glow green under UV light, right? But we are going to give them the genetic information to do that. So when you shine UV light on them, they glow. They've been transformed, right? And that's a pretty big change, right? From not glowing to glowing. And I'll bring them in so you guys can see glowing E. coli too when we make them in lab. Right? You can't take them home. I've had students in my lab like, can I take this home? No. Cannot take this home. This cannot be out of a lab setting. But there's a reason why we do that. It's not just for fun. It's a good genetic tag, just like how I talked about antibiotic resistance, helping know that my experiment worked. It's a good way of being able to tag an organism. I mean, they've done it to organism levels. They have glowing cats and glowing mice, and that's because they've genetically altered them, and they know it worked because if it didn't, they wouldn't glow, right? So they've transformed them. That's just a tag. There's a really cool video link that I'll put for you guys. They did an experiment where they genetically altered fruit fly sperm. So one fruit fly sperm was fluorescent green and one's fluorescent red. And female fruit flies mate with more than one male so that her offspring are genetically diverse. But the males, of course, want to be the winners, right? They're the only ones that want to fertilize the egg. So in the female reproductive tract, since they were able to color the two different sperm different colors, you actually see there's competition between the sperm to get to the egg. They're like, no, 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 those are my eggs, right? And they filmed it. And the only reason why you could see and know that they're different is because they genetically labeled them, made them different colors, right? That experiment would not at all be possible if they didn't do that transformation of those sperm. I show that to my kids over the summer, the high school and junior high STEM kids. They love it. You say sperm, they pay attention all the time, right? So do you guys. Right. So I'll post that link for you. It's just a short little video. It's kind of cool, but you can see the sperm, the two different colored ones, right, swimming and trying to get past each other. So transformation, can you guys imagine that now, right? Something transforming, right? Major change. Transduction, focus in on the duct part of this word. A duct is something, what things travel through, right? So in this case, the duct is going to be a virus. The DNA is going to be transmitted from one bacteria to another by a virus. So that duct, right, is the virus, right? So when you think transduction, think of that duct. Think of viruses being that conduit that moves stuff from one thing to another. And then hopefully you'll never get these two confused. So in order for this to happen, though, you have to have a donor cell, all right, that's going to accept stuff from a recipient, right? So stuff is going from one cell to another. The donor cell, right, has these three different ways of moving genetic information from it to its recipient. Once the recipient takes this information, it actually has to utilize it to be either transformed, transduced, or conjugated. So probably the best example of this is, imagine you're walking down the hallway and you see $1 on the ground. Unless you saw someone drop it, right, it's yours. Finders, keepers, losers, weepers, right? Money doesn't have name tags on it. Cash money. Right. It's another reason why I don't like to carry it when I don't have to. So if I see it walking down the hall, I'm probably going right straight to the vending machine. Right? Get me something out of the vending machine. So, you know, I've been transformed, right, because I took something from the environment and I utilized it. Right? I went that second step. So you need to understand the steps of the process specifically for DNA-mediated transformation. So it starts out with somebody dying, unfortunately. Some bacteria dies and its DNA becomes part of the environment. So that's what's happening here. Here's a bacterial cell. It's dying. Its DNA is rupturing out of its cell. All these lines you see here are its DNA. So it's like that person walking down the hallway drops the $1. Right? In this case, the bacteria dies and drops his DNA into the environment. So it's up for grabs, right? Literally, anybody can grab onto it and take it. And that's what happens. Right? So again, remember there are animations, right, that help out for some people. I'm going to skip over that. But if you're in presentation mode, it will play. So this phenomenon, this natural phenomenon that happens bacteria, was actually discovered by Frederick Griffith in 1928. And he was working with Streptococcus pneumoniae. And he was actually trying to develop a vaccine. He wasn't trying to discover DNA-mediated transformation. But he did. Oh, I don't have the diagram for it. That stinks. Okay. I'll add a diagram for you guys. I like pictures. Lots of us learn better with pictures. So what he was doing, as I said, was trying to develop a vaccine. And in doing that, he was treating bacteria, and there's actually 2 different strains of Streptococcus pneumoniae. One strain has a capsule, and one strain does not. So one looks kind of smooth, the capsulated strain, and the other one is kind of rough, that doesn't have a capsule. And he found that this strain that was kind of rough, it didn't cause pneumonia in the animals. And we know the reasoning behind that now is because it doesn't have a capsule. Our immune system was able to recognize it and destroy it really quickly. The encapsulated strains of Streptococcus pneumoniae, on the other hand, that capsule helps them adhere to us and helps them evade our immune system. So that causes a very serious form of pneumonia that can lead to death. And so he was, of course, experimenting on mice. And one of the things that he did was that he heat killed the encapsulated strain and gave it to the mice. Well, of course, the bacteria are dead, so the mice aren't going to die. He also found that, of course, when he gave them the rough strain that don't have the capsule, even alive, he could give this to the mice and they would survive. So one thing is good, why not 2 things, right? Why not combine the 2? So that's what he did. He took the living strain of the rough bacteria that didn't kill the mice and the dead ones that do have the capsule that do kill the mice when it's alive, but not when it's dead. He combined those 2 and put it into the mouse. Well, the problem with that is that the pathogenic encapsulated strain, how does it know how to make that capsule? It's in its DNA. So now by putting those cells, even though they're dead, into the same environment as the living cells, those living cells are able to pick up the DNA from the dead cells. And that's what they did. And they picked up the information on how to make a capsule and started making a capsule. So lo and behold, the mouse dies. That's not cool. And then he autopsies the mouse and finds this bacteria living with a capsule. How did that happen? He didn't put living encapsulated bacteria in there. No, but he put DNA in there on how to make a capsule, and they utilized it. Right? So they transformed by picking up DNA from the environment. And in this case, it was very advantageous for that bacteria learned how to make a capsule from one of its close relatives, another strain of the same species. That, of course, would not work for a vaccine for us, right? Instead, they give you pieces of bacteria or pieces of the capsule so that you make antibodies against it so that your immune system recognizes it. They don't give you living stuff. They'll give you weakened forms of bacteria. They're not going to give you the big bad bug, right, in vaccines. And give your immune system information in a safe as possible way so that you recognize it through vaccination as opposed to natural exposure. Gives your immune system a one-up. So, right, he accidentally discovered, right, what was going on, right? What went awry, you could say, with his experiment of trying to develop a vaccine. So what happened? This involves a transfer of naked DNA from the environment to the recipient. How did that stuff get into the environment? A cell died, right? During stationary and death phase, as you saw in that picture, the chromosome will break into small pieces. It explodes through the ruptured cell wall. The recipient cell comes along. It's going to pick up the pieces of naked DNA. And it actually, it'll be double-stranded, but they'll only bring in a single-stranded. And again, only cells under certain conditions are going to do this. But they have receptors on their surface under certain conditions to be able to pick up DNA from the environment. They'll bring it in single-stranded, and they'll see if there's some place they want to stick it in their chromosome, right? Do I want to switch this out for something else that I'm not using, right? And they're going to add it in, right? That's that homologous gene, right? Where can I put this? I don't know, honestly, if they know what they're picking up. We don't understand them to quite that level. I mean, of course, they're not like us. They don't think about things. But is there some mechanism to understand that this DNA is good as opposed to bad? But the thing is that sometimes they'll take stuff in and they won't incorporate it. They don't, like, find a place to put it, right? And so they're not truly transformed unless they actually incorporate it. Excellent question, Devante. So as I said, it's under only certain conditions even that they'll do this, and we call this competency. And we can purposely stress bacteria to make them competent. We can stress them. We can use electroporation. We can use electricity to infuse DNA into cells. So we can cause this to happen in a laboratory situation. We can make them chemically competent. Mostly in nature, it's when they're under stressful conditions. When food is becoming scarce, right? When they're under a condition where it may be beneficial to acquire some other genetic information. Did you have a question?
I did. I wanted to know if it was even possible to pick up something that could possibly kill the living cell.
Be harmful to them. I would say it probably is possible that they could pick up something that was harmful and that it would hurt them. But again, I would, like I said, we don't understand it to that level of detail currently in our research. But I think maybe they might have the ability to detect if it's something they don't want. Because there are times where they won't take stuff in, where it won't work. So they're only truly transformed if all the steps of the process happen, right? They acquire the DNA, they incorporate it into their genome, and then later they can pass it vertically onto their progeny, right? Once they have it, right? And they pass it on to all their progeny. So once, that strain of E. coli incorporated it, right, all their progeny have that ability now of producing that toxin. And then notice non-transformed, right? They didn't do it. So for transduction, we start out with a bacteria being infected by a virus. Bacteria have their own viruses. They don't infect our cells. Their cells are distinctly different from ours. Their viruses have to be different, therefore, to be able to infect them. And we'll talk about that a little bit later. So, in order to be able to infect, they got to be able to bind to the bacteria. And the way bacterial phage or bacterial viruses work is they actually inject their genetic information into the cell. Because of that cell wall, the whole virus doesn't enter. There's two types of transduction. Any gene from a donor can be transferred, where specialized, as the name implies, only special genes can be transferred. So as I said, this could be quite a complicated process. We're going to keep it at the lower level. So what happens is the cell gets infected by a bacterial phage. A bacterial cell gets infected. The bacterial phage genetic information gets injected into the cell. And now they're going to take over the cell, right? This virus is going to take over the cell. It's going to have that cell make copies of it, of that virus, of its protein code, of its genetic information. So you'll notice that it's coded, and it'll destroy the host cell's DNA because it doesn't want it to be a cell anymore. It wants to be a virus-making machine factory. But when they chew up the host cell's DNA, sometimes it gets accidentally packaged into the viral code of the virus. So notice blue denotes bacterial DNA. So in this case, this one virus right here picked up the wrong stuff. It's not viral DNA, it's bacterial DNA. Yeah, it's kind of an ineffective virus because it doesn't have the information it needs. So now when it goes and infects another cell, that cell doesn't become a virus-making factory because the information there is not there on how to make the virus. It's just transmitting information from one bacterial cell to another, right? It's acting as a vehicle, as a duct. So this is a defective virus, right? By making this mistake, it just became a free carrier for bacterial DNA, and its life cycle is going to end, right? So lots of things, quote-unquote, die in this. The donor cell, where the DNA originally came from, died due to a virus infection. And this virus, you could say, quote-unquote, died by picking up bacterial DNA instead of viral DNA. So all it is a carrier to another cell. When this stuff is injected into that cell, if the cell incorporates it, it's now been transduced. It's been changed, right? Information has been transferred to it via transduction of virus, making a mistake. Even viruses make mistakes. In this case, the bacteria benefits, right? Because it just got, instead of a viral infection, it just got, hey, here's some bacteria from one of your, here's some DNA from one of your bacterial friends. You want to use it? Okay. So we've probably introduced plasmids and bacterial chromosomes previously, but it's a good chance to point out these differences of sources of DNA within bacterial cells. So not all cells have plasmids, but they're very common in bacteria. And they're smaller than the chromosome, and they code for non-essential information. So a bacteria cell doesn't have to have it. But it is DNA. Right? It's smaller, it's non-essential. But if they do have it, it can give them extra abilities. And they frequently will have more copies of this plasmid than they will the chromosome. When a bacteria cell copies its chromosome, it's usually because the cell's getting ready to divide. Where for plasmids, for some reason, it'll keep numerous copies. Which is great for scientists like myself when we're using bacteria to make copies of DNA for us. They'll make a whole bunch of copies of a plasmid for us, and we can just lyse the cells and purify out the plasmid and use it for our research. This is done a lot. Nobody cares about bacteria. They're not cute and fuzzy. They're pretty awesome. They do some really cool things for our life nowadays that just weren't possible before these types of technologies. So resistant plasmids are bad news for us, and of course great news for bacteria. These are specialized plasmids, and they're called R plasmids, which stands for resistance. And so these are plasmids that exist in nature that contain genes on them for antibiotic resistance, right? Whether it's making an enzyme or making something else for that cell to function without the antibiotics affecting them anymore. So this particular plasmid actually has 3 different resistant genes on it for 3 different antibiotics. You have chloramphenicol, cambriocillin, and tetracycline. That would really not be good if a bacteria you're infected with had that level of resistance. So one of the most common ways we believe large pieces of DNA, like these plasmids, these circular pieces of DNA, are moving is through conjugation. And so a cell that has a particular plasmid is said to be F positive. And they would denote what plasmid or what genetic information that organism actually has. A recipient cell that doesn't have this information will be referred to as negative. F standing for fertility, the ability to share information, to have information. So positive is having particular information, negative is not having it. And this can be right down to the genetic level. So you can have cells that are F positive for one gene and F negative for another. The same cell. So, as I said, very common in this, of course, especially when they're sharing things like resistant plasmids. This is of great concern for those of us trying to treat bacterial infections, right? And so this is one of the main ways they believe this information is moving is via conjugation. So that process is where you have a donor cell, right, that has the plasmid, so it would be called F positive, and a recipient cell who's F negative who doesn't have it. So this is the only form of gene transfer where both of them get to live, right? The donor lives through the process, the recipient lives through the process. So these guys are going to join up with each other, and through that sex papillae or the fertility papillae, a hollow tube, they're going to connect to each other. So they directly connect with each other. So here's a picture of that, right? So there's that tube connecting these 2 bacteria. Now we don't know which way the information is traveling. We don't know who the donor or the recipient is in this picture. We just know they're conjugating, right? Because they have the sex or fertility or F bacillus connecting them. So they have that contact. The genetic information is going to move from one to another, in this case in the form of a plasmid. And then that recipient is going to utilize and keep that plasmid. So diagrammically now, this guy has changed too. He's got the same ability as his friend. So now the donor, right, gave to the recipient, but then this recipient becomes a donor. It can go and share that information to other cells. So you stick one bacteria in a test tube that has information. If he can, he's going to give it to all his buddies in that test tube. They're going to share that information. So remember, one of the main reasons why we talk about this genetics is, as we said, this can lead to antibiotic resistance, right? Positive, good change in that level is not good for us in the allied health fields.